Okay, so in the next couple of sections, we're going to be talking about cellular and humoral immunity in a bit more detail than we already have in the last section. So starting with cellular immunity, this is again the form of adaptive immunity that uses T lymphocytes to attack or destroy cells, being foreign cells or diseased host cells, directly. Okay, so this type of immunity allows your immune system to remember antigens or other types of pathogens and invaders in order to prevent them from causing disease in the future if you have some sort of secondary encounter. There are 4 classes of T cells that are employed in cellular immunity. So cytotoxic T cells are the first and probably, I guess most important that we'll talk about. They're the ones that carry out the attack on foreign cells directly. So here we have cytotoxic T cells attacking a cancer cell. Okay, so again, they carry out the attack of on foreign cells. Helper T cells, on the other hand, help the activity of other T cells by releasing cytokines. So if we remember cytokines from our last section, they're chemicals that our lymphocytes or other leukocytes use to communicate with each other. Okay, and so these helper T cells basically will bind to an antigen or an antigen presenting cell and release these cytokines or chemicals that will communicate with other leukocytes or other lymphocytes to bring them into this site and activate them in some way. So helper T cells are going to be important. This doesn't really matter to you yet, but it will in the next set of slides that we go through in the next section of this chapter. But helper T cells are involved in both humoral and cellular immunity. So we'll talk about their involvement in each as we move through. Regulatory T cells, we don't spend much time talking about those, but they are important because they're what are responsible for limiting the immune response after a pathogen has been defeated. So without these guys, maybe the T cells just continue multiplying and we continue to secrete these inflammatory cytokines, which could have really extensive damage effects on our tissues in the long term. So regulatory T cells are really important for making sure that that doesn't happen. And then finally, memory T cells are responsible for memory and cellular immunity. So how do we remember a pathogen the next time that we come into contact with it, or an antigen? So there are 3 R's in both humoral and cellular immunity, but today we're going to be, or at least in this section, we're going to be focusing on just the steps of cellular immunity. So the 3 R's are recognize, react, which basically just means attack, and remember, or memory. And within cellular immunity, recognize occurs in 2 steps that we've sort of brushed on already: antigen presentation and then T cell activation. So that's what we're going to focus on first. The first step of cellular immunity, which we just decided is recognition, and then that first part of recognition, which is antigen presentation. So we've already talked about this in the last section of the chapter, but basically, again, what happens is this big antigen presenting cell here comes into contact with an antigen. It's going to bring that antigen into the cell, digest it, chop it up into little pieces, display the epitopes, or the active part of the antigen, on the surface of the antigen presenting cell at what's called this MHC protein. From here, that antigen presenting cell is going to migrate to a lymph node where it will come into contact with either helper T cells or cytotoxic T cells. So this macrophage here is acting as an antigen presenting cell, and it has an epitope on an MHC protein at its surface, which you can't see because this helper T cell is bound to that epitope at the MHC protein. Okay, so when this T cell encounters a cell that is presenting an antigen, like this macrophage here, then it'll bind and initiate an immune response. So that's the first part of recognition, is antigen presentation. The second part of recognition in cellular immunity is T cell activation. So we already said all of this stuff happens. The antigen-presenting cell takes in an antigen, presents it on the surface. It's going to bind to a helper T cell, and then that T cell will be activated. So a helper T cell or a cytotoxic T cell, which remember is the type of T cell that directly attacks an antigen, will bind to this MHC protein here on the antigen-presenting cell that's displaying the epitope. Okay, so the first step is the antigen is presented here. The second step is that the T cell binds and is activated or starts some sort of immune response. So we're going to kind of skip ahead a little bit to the second steps, and then we'll reiterate them. So this is really this is really just your first two parts. This is sort of getting into the second step, which is the attack. It's sort of an in-between step. So basically what happens here is what we call co-stimulation. We get this T cell, either a helper T cell or a cytotoxic T cell that binds to the antigen-presenting cell at the MHC protein through this epitope of the antigen. And in order for this reaction to go any further, we have to have what we call co-stimulation. So we have this second protein over here called a co-stimulation protein. And so your T cells basically have to bind not only to this epitope, but also to this co-stimulation protein. And this is just basically a mechanism for this T cell right here to basically double check that it's bound to an antigen-presenting cell that is in fact displaying a suspicious antigen. So if this co-stimulation doesn't occur, we don't get a binding here and then also here, then the T cell right here will become inactive. If co-stimulation does occur, then this T cell right here is going to bind to that co-stimulation protein and we'll get clonal selection. Okay, so basically this T cell is going to be cloned through repeated mitosis to create identical T cells that all target this same epitope. So some of those T cells will carry out the attack, like these guys. I mean, cytotoxic T cells are the only ones that really carry out an attack, but helper T cells are involved in that process. And then also we get these memory T cells. Okay, so we sort of skipped ahead a little bit, but that is an overall recap of the second step of cellular immunity, which is the attack itself. So again, these helper T cells, there's two different scenarios that can happen. We can have helper T cells that recognize the MHC protein complex containing the epitope of the antigen on the antigen-presenting cell, and as a result will secrete these interleukins. So these cytokines, which again are chemicals that the cells use to communicate with each other. And the effect of these interleukins that we see here are that will attract neutrophils, natural killer cells, macrophages, but they also stimulate B and T cells to reproduce and also to mature. Okay, so these interleukins communicate with a lot of other different leukocytes. The second type of reaction that can happen is that we get a cytotoxic T cell that binds directly to the enemy cell. And remember, cytotoxic T cells are the only T cells that directly attack a pathogen or an antigen. So what they're going to do is they're going to dock to this enemy cell, okay, and basically deliver a lethal hit of chemicals to destroy it. So that lethal hit of chemicals could be in the form of porphyrins and granzymes, which we talked about in innate immunity. If you remember, they're little proteins that basically drill a hole in the side of the cell, and then it releases these granzymes to penetrate the cell and destroy all of the enzymes inside of the cell. So that's one way that the cell can die. Also, maybe it secretes interferons, which will inhibit the replication of any virus, or tumor necrosis factors, which are going to attack cancer cells. So that's what you're seeing here. You have a dying cancer cell and all of these cytotoxic T cells acting on it. In the third and final step of cellular immunity, we have memory. So that last R, which is remember. So we talked about how if we get co-stimulation here, meaning the T cell binds to the epitope on the MHC protein of the antigen-presenting cell, and also to the co-stimulating protein here, we get cloning. And so the important part of this cloning in memory is that some of those cells are going to become memory T cells. And these are really long-lived cells in our bodies. So that next time we're exposed to the same exact antigen, which we have right here, we don't have to wait for the production of all of these cells to happen and all of this cloning to happen. We already have a large number of these memory cells. So that's going to destroy a pathogen really quickly, and most of the time before we even notice that we've come into contact with it. So maybe we'll never even get quote-unquote sick, or we'll never have any clinical symptoms of the illness. Okay, so practice questions for this section. What are the 4 types of T lymphocytes? We talked about cytotoxic T cells that are responsible for the attack itself, helper T cells, which release cytokines or chemicals that allow it to communicate with a whole bunch of other leukocytes to target a pathogen. We talked about regulatory T cells. What do they do? Why are they important? And then memory T cells. Which lymphocyte is essential to humoral immunity? So again, this wasn't necessarily important for this section, but it will be for the next section. So I just want you guys to remember that helper T cells are involved in cellular immunity and also involved in humoral immunity. So remember, cellular immunity is the type of immunity where you get T lymphocytes that target and attack a pathogen directly or an antigen directly. Humoral immunity is where we use antibodies instead. So that's what we're going to talk about in the next section. And then what are the 3 phases of an immune response? What are the 3 R's? We have recognition, we have react, and then we have remember. Okay, so in cellular immunity, there are 2 parts to recognition: antigen presentation and then T cell activation. So talk about each of those. And then in the reaction phase, what's happening there? There are 2 different ways that a cell could react. We could employ helper T cells, or we can employ cytotoxic T cells directly. And then finally, remember. How do our cells remember for the next time that we come into contact with an antigen? And then finally, describe some ways in which cytotoxic T cells destroy target cells. We talked about perforins and granzymes. What do those do? We talked about tumor necrosis factors. We talked about one other thing here. Oh, interferons. Okay, so talk about what each of those and what their functions are. All right, so now we're going to talk about humoral immunity, and hopefully we're comfortable with cellular immunity, and we can make the contrast between the 2 as we move through these slides. So within humoral immunity, again, distinguishable from cellular immunity, in this type of immunity, we do not employ lymphocytes that directly attack enemy cells. Okay, B lymphocytes are used in humoral immunity as opposed to T lymphocytes, and they make antibodies, which we see right here. So here's a lymphocyte producing antibodies, and those antibodies are going to bind to the antigens. So you see the same thing down here, a lymphocyte making antibodies and binding to an antigen. But those antibodies don't necessarily destroy the antigen themselves. They can neutralize the antigen, which we'll talk about as we move through the slides, but they basically tag them for destruction through other means. Okay, and we have the same 3 stages of humoral immunity as we did with cellular: in recognition, react or attack, and remember or memory. So in the first step of humoral immunity, which is recognition, what happens is we get antigens that bind to receptors on a B cell. So that's where you see all these little green dots here are binding to these receptors on the B cell. And those antigens get brought into the cell like this. So here we have all these receptors on the surface. The antigens are binding to the receptors. The receptors then get sort of linked together like this, and the antigens then come into the cell through endocytosis. Okay, so that's what's happening here. You don't really see it very well in this image, which is why I wanted to provide you with this one. But basically, from here, the antigens are brought into the cell through endocytosis, like we see up here. Once that happens and the B cell has brought the antigen into the cell, it's going to process it, digest it, and act as an antigen-presenting cell. So it digests the antigens, chops it up into little tiny pieces, and then exposes the epitope, or the active part of the antigen, on this MHC surface protein. Okay, and what that does is it presents the antigen to helper T cells, which we had already seen in cellular immunity. They're the only T cell that are also going to be involved in humoral immunity. So that helper T cell is going to bind to this complex and secrete interferons, which are cytokines or chemicals that are used for these cells to communicate with each other. Upon the release of these interferons, this B cell is going to be stimulated to clone itself. Okay, and then from there, the majority of those cloned B cells will become plasma cells. And some of them will become memory cells, but the majority will become plasma cells. So again, let's go through this one more time. Recognition, basically an antigen binds to the surface of the B cell. It's brought into the cell through endocytosis. The antigen, once inside the cell, gets chopped up into little tiny pieces and exposed. The epitope gets exposed on the MHC protein of the B cell. What that does is it presents the epitope to these helper T cells, which will bind and secrete interleukins. That interleukin will stimulate the production of more B cells, or cloning, and the majority of those B cells will turn into plasma cells. What are plasma cells? Why are they important? They're produced from cloned B cells, and we have them down here. The majority of them are going to live in the bone marrow until they die, and they'll live for a period of 4 to 5 days, but their job during that 4 to 5 days of life is to pump out antibodies. So that's what you see here, and they're very efficient at doing this. They make about 2,000 molecules per second, and then these antibodies that they produce will be circulated through the blood and other fluids. Okay, so plasma cells come from cloned B cells, and their job is to produce antibodies. Alright, so in the second step of humoral immunity, we have react or attack. So we've said this before, in humoral immunity, we don't have T lymphocytes that are directly attacking cells. We have B cells that produce antibodies, and those antibodies themselves do not harm an antigen. They can neutralize an antigen, or they can mark that antigen for destruction by other agents, such as macrophages, eosinophils, or complement proteins. Okay, so they're just acting as little markers. And there are 4 mechanisms of antibodies to target antigens. So we'll talk about each of these 4 in more detail. We get neutralization of antigens through antibodies. So what happens here is, in this part of the picture, you're seeing a normal antigen binding to a host cell. But what happens is these antibodies will first bind to the antigen, so now their binding sites are blocked and they're not able to bind to the host cell. So they get kicked off here. Complement fixation is the next way that antibodies combat antigens. Basically, these antibodies are going to bind to the antigens and also bind to complement proteins. So if you remember what complement proteins are, they're part of the innate immune response. We talked about them last time, and so they will bind to these cells and destroy them. And they do that through inflammation, phagocytosis, immune clearance, and cytolysis, which are 4 things that we talked about in the last lecture. So this is the primary mechanism that our body uses against bacteria and also mismatched blood types. So that's why in this one we have a red blood cell here. So you have your antigens on the surface of red blood cells. They're bound to antibodies, which then bind complement proteins, and then the complement proteins themselves are what employ the destruction of the red blood cell, not the antibodies themselves. Agglutination, we talked about this when we talked about blood cells as well. It's just the clumping of a bunch of enemy cells by a single antibody. So these antibodies here we see in green have anywhere from 2 to 10 binding sites, and so they can bind more than one antigen at a time. And so what that does is, in this case, we have red blood cells as our antigen. That they would be like a foreign red blood cell. And so what that does is it immobilizes pathogens to keep them from spreading because they're all clumped together, and it allows for macrophages and other phagocytes to more efficiently consume enemy cells. So if we have like a bunch of these stuck together, let's just say we have 3 of them stuck together, consuming or having a macrophage consume 3 enemy cells as opposed to 1 is just a more efficient means of phagocytosis. So this type of means of an antibody combating a pathogen is effective against bacteria and also mismatched blood types, just like complement fixation. And then finally we have precipitation, which is much like agglutination, except it involves the clumping and adhesion of free antigen molecules. So remember we talked about venoms are an example of free antigen molecules as opposed to molecules on the surface of a bacteria. So we basically create all of these antibody-antigen complexes that are removed from the body through immune clearance, and they can also be phagocytized by eosinophils. So immune clearance is something we talked about in the last lecture. Okay, so those are our first 2 steps. The first was recognition. So again, an antigen comes into the cell, into the B cell, it's chopped up, the epitope is exposed on the MHC protein, and then we get cloning, which those B cells turn into plasma cells. So that's recognition. The react portion of that is the neutralization. So we don't allow an antigen to bind to a host cell. Also complement fixation. The cell is destroyed by complement proteins. Agglutination and precipitation, which is just the clumping of molecules or clumping of free antigens. So that's the attack portion. And then the last portion of humoral immunity is the memory part. So during a primary response, which a primary response is just the immune reaction that you have when you're exposed to a particular antigen for the very first time. This whole process that we see here of exposing to the antigen, bringing it into the cell, presenting it, cloning those B cells, and then differentiating those B cells into plasma cells and, of course, memory cells, that takes, well, and then production of antibodies from those plasma cells, that takes about 3 to 6 days. Okay, so that's what we see here, 3 to 6 days for the primary response in order to get those naive B cells. So what happens is we see that at about 6 days after exposure to an antigen, our body starts to produce antibodies. It'll peak and it'll come back down and drop back to normal within about a month or so. During clonal selection at that first exposure, we saw that some of those clone cells become memory cells. So let's go back up here. We get a bunch of plasma cells which create antibodies, but we also get memory cells. And so those memory cells are important because they mount a really quick secondary response if we're exposed to the same antigen again. Okay, so here's the primary response that we just looked at, peaks at 6 days of exposure, and then comes back down within 30 days. With the secondary response, we get a way quicker peak of, or rising of the antibody titers. So those are going to rise really sharply, and then they're going to stay elevated for much longer periods of time. So sometimes it's just weeks, but sometimes it can be years. And this occurs so rapidly that, again, we can see on day 0, we have rapid production of those antibodies. So we really have very little time for the antigen to exert any noticeable effect on the body. We may not even realize that we've come into contact with the antigen. All right, so practice questions for this section. Distinguish between a B cell and a plasma cell, right, because they're not the same thing. One comes from the other. Here we have B cells. Here we have plasma cells. What do plasma cells do? Describe 4 ways in which an antibody acts against an antigen. So we talked about neutralization, right? We block a pathogen from binding to a host cell. We talked about complement fixation, so these complement proteins were going to bind to the antibodies, which then bind to the foreign cell and actually do the destruction themselves. And again, if you want more information on complement fixation or complement proteins, that is part of innate immunity, so go ahead back to the last lecture and brush up on that. Agglutination of antigens and precipitation, which is just sort of clumping of either these enemy cells or the free-floating antigen molecules themselves. And then why does the secondary immune response prevent a pathogen from causing disease while the primary response does not? Right? So during this primary response, we get cloning. What are the results of this cloning process? We have plasma cells, but we also have these memory cells. So talk about their role and why we get this curve during the first response and then this curve during the second response. Alright, so I'm going to terminate this video here just so it uploads to YouTube faster, and then we have one more section on immunodeficiency diseases and different things like that.